Thank you. And thank you, Dr. Doudna, for joining us today. Genomic engineering has been around for decades now. What makes CRISPR-Cas9 so different?
The CRISPR technology is a relatively new way to make precise changes to the DNA in cells and organisms. And what makes it different than earlier technologies is that it's simpler and easier to use. So I like to call it a democratizing technology. It makes precision genome editing available to labs around the world that have a skill in molecular biology.
So in the beginning, you discovered this technology in bacterial immune responses to viruses. So when did you realize that you could apply this to the human genome?
I think the wonderful backstory to CRISPR is that it's a technology that came out of an effort to understand a fundamental process in biology, which is how bacteria fight viral infection. And I remember the moment actually in the laboratory working with Martin Janek in my lab, who was purifying the protein called Cas9 and figured out that it operates as essentially a molecular scalpel. It's a way to cut DNA at precise places. And the thing that makes it so powerful is that it can cut DNA in a programmed way. So we can program it, kind of analogous to the way you would program your computer to do something. And so Martin had done an experiment showing that he could program the Cas9 protein to find and cut a series of different DNA sequences. And it really, at that moment, we recognized that this could be harnessed for a different purpose, namely for gene editing in other kinds of cells.
So you mentioned how CRISPR technology works in bacteria. Does it work the same way in humans?
It does. It works the same way in humans. So it's able to find and cut DNA sequences that match the sequence of the RNA molecule that it's programmed with.
CRISPR technology literally has the ability to change the genetic code of a human life. For someone who's considering this technology, what could go wrong?
Well, lots of things could go wrong. The change could have an unintended consequence. That would be one possibility. Also, the change could occur at an undesired place. So there could be a sort of so-called off-target editing that occurs. That's sort of collateral damage, you could call it, I guess. So there's that possibility. I think it's going to be very hard to figure out how to do any clinical testing on such a thing, right? How would you actually test that on people without using it on people? So I think that's one of the challenges as well, is that I think we'll have to proceed very carefully using this in ways that are where there's going to be very clearly a big benefit versus the risk in the early days. And then as the technology advances, it may be possible to use it for other things where again, I think for every application we're going to have to balance the risk versus the benefit to decide if it's worth doing.
So how could the CRISPR technology impact future generations?
Well, I think it's important to make a distinction here. So when we talk about, we can talk about two different kinds of editing, gene editing that could be done in human beings. One would be what we call somatic cell editing. So that means making changes to the DNA of cells in a person in a way that doesn't allow those changes to be passed on to future generations. But the other kind of editing would be changes that would happen in the germline. And that means changes that could be passed to future children of that person and their kids, et cetera. And so then, we could imagine very long-lasting impacts of making a change, which is, I think, one of the reasons why doing that kind of editing has to be very carefully handled. And so that will lead to an ethical question, right? Do we do that where it's easier to do and where you avoid someone ever getting a genetic disease? Or do we decide that's not something appropriate to do? And I think that's one of the challenges that we currently face.
Right now, there's a lot of speculation on what it could do. We want to know realistically, in the next 5, 10, 20 years, what it can do.
We published our work in 2012, and we're now in 2017, so it's only a 5-year-old technology. And this is sort of a remarkable thing to see how fast it's been adopted for various applications. And right now, there are already clinical trials that are starting using the CRISPR technology to treat cancer patients. So that's the first step. But I'm thinking in the next probably 1 to 2 years, we will see clinical trials beginning for treating genetic diseases like sickle cell disease, which is a blood disorder that has a well-known genetic cause and has already been corrected using the CRISPR-Cas technology in the laboratory. So we're now moving to a stage where it could begin to be tested in a clinical setting. And then beyond that, I think, if we talk sort of maybe 5 to 10 years out, we'll start seeing clinical trials for other genetic diseases such as muscular dystrophy and possibly even cystic fibrosis. So I think these are examples where we understand the genetic basis. There's a single mutation that could be corrected, and this technology is primed to do that.
So you already mentioned that there has been research done on sickle cell and cystic fibrosis. How do you prioritize the diseases that are researched?
Well, I think, the thing about the CRISPR technology is that I think it will be most useful initially in the clinic for treating diseases that have a well-known single genetic cause, a single gene that has a mutation. But as you know, many diseases have, a multitude of genetic causes. And so I think those will be obviously a lot more challenging to treat with gene editing. Nonetheless, I think that for those kinds of diseases, we're seeing a real increase in the pace of research to understand the genetic basis. And then that, hopefully in the future, will lead to opportunities for real treatments as well.
When do you think it will be applied to more genetically complex diseases?
Yeah, it's hard to say. I mean, I guess there I'm thinking we're probably looking out a decade or so. I could be wrong because I've been amazed at how fast the pace of research has been moving over the last few years. So certainly things are moving quickly, but I do think that it'll be a lot more challenging to treat diseases that have multiple genetic defects.
Can you point to any public policies, either current or prospective, that are relevant to the development of this technology?
Yes. We're fortunate that there actually is a very nice framework in place for managing and regulating the use of technologies like the CRISPR gene editing tool. Part of this comes from the work that was done back in the 1970s in a meeting that's known kind of in the scientific community as the Asilomar meeting that happened back in 1975 to discuss molecular cloning. And that meeting led to a series of discussions and eventually regulations put in place to monitor and guide the use of technology that would allow DNA to be copied and to be put into other organisms. And so we're able to operate under the same guidelines for this technology as well. So that's great. And the ongoing discussions right now are really about asking, in the future, are there going to be uses of this technology that will require additional regulation? And that's part of very much part of an ongoing discussion.
What commercial application of CRISPR are you most excited to see?
Well, I think, again, this gets back to what we talked about earlier with, in terms of thinking about where it's going to have the biggest impact. And I do think that when we think about agriculture, this is an interesting thing where, we can think about an impact on many, many people that avoids the whole question of, access to insurance and things like that, right? If we have ways of developing plants that have higher nutritional value or can grow better in certain environments, I think that's something that could be extremely impactful on human health. And so that research and technology is also moving ahead very quickly, and I'm personally excited about the opportunities there as well.
CRISPR has the ability to change certain aspects of people's lives, such as preventing allergies or color blindness. Where do you think the line should be drawn between life-saving aspects of CRISPR and life-changing uses?
Well, for myself personally, I think that I'd like to see this technology deployed initially, at least, to understand the human condition better, understand our own genomes. And remarkably, we still don't understand that much about the information that's encoded in our own DNA. So this is a tool that will clearly going to accelerate that. And then beyond that, I think in terms of thinking about applications across different conditions that will affect human beings, I personally would like to see it used to help treat or cure genetic diseases, things that we talked about earlier, I don't think that the time is right anytime soon to use it for other purposes. I think we will have to really understand how such changes will affect not only a person that might receive such a treatment, but also how it affects their sort of family and future generations, and thinking about the societal impact of that kind of application. So I have really spoken out publicly about refraining from that kind of use of this technology until we have a chance to fully discuss it publicly and educate people about this opportunity and frankly, challenge.
You mentioned that you don't believe people should go there, but many people have been discussing and associating the CRISPR technology with designer babies and super athletes. How legitimate are their concerns?
Well, I think they're legitimate, because I think we're now really for the first time in human history, human beings have the ability to alter their own DNA in a way that could affect human evolution. I mean, it's kind of really profound to think about that. And even though there are practical restraints on that we've been discussing, it's a very real possibility now in the not distant future. So this is why I think it's critical to be, educating the public about this, encouraging people to think about it and to get involved. That this shouldn't be something going on behind the walls of research laboratories, but it really should be something that all of us as part of the human community should be discussing, because it really belongs to everyone.
Is there another application of CRISPR that you feel we missed? As the inventor, is there a certain thing that you think people aren't applying CRISPR for that it should be applied?
Boy, that's a tough one, because it's being used so widely. But there's one sort of broad use that we didn't talk about yet, and that is in the research community. So one of the things that for me has been very exciting is that the genome editing now has opened the door to doing research and experiments in organisms that in the past were basically genetically intractable. And so I get, pretty regularly, I get emails mostly from students that are working in research labs, and they share some of their data with me. And so recently I got an email from somebody that was working in butterflies, and was in the past was only able to observe butterflies and wasn't really able to do any actual manipulation of butterflies. But now with gene editing, he's able to change the genes that alter the butterfly wing patterns, and so they can really study that process at a much more detailed level. So I think that's something that people maybe don't appreciate, is that it's really a technology that has increased the pace of research just across the board in biology.
One more question. What criteria will you use to decide whether or not others can license your patent?
Well, it's not up to me, actually, because the work that we did is owned by the University of California. So the University of California, as a public university, is responsible for licensing those patent rights and deciding which commercial operations will get access to the technology. But I do want to point out that the technology is freely available to anybody that's working in academia. So anybody doing not-for-profit research is freely able to access this, and I think that's one of the things that makes it so exciting, is that we just see it being adopted globally, really, for all sorts of applications in research and technology.
Great. Thank you, Dr. Doudna, for joining us today and for your expertise.
It's been a pleasure to be here.
Back to you in the studio.